Okay, so here's the DNA sequence that was given to us in prompt 41B, and I have translated it and determined the reading frame and got this corresponding amino acid sequence. So we're going to go through all the point mutations that could possibly happen to this sequence, and we're going to start with missense. So a missense mutation changes the amino acid, so it changes it from one to the other, and we're going to do that with AAC, and we're going to change that into AAA, which is changing a pyrimidine to a purine, so that will be a transversion. So what we're going to do is we're going to switch this C for an A, and instead of getting ANS, we're going to get a lysine instead, and that missense mutation could make the protein non-functional, or it could just change the function of it. So going back to our original sequence, the next type of mutation we can go through is a silent mutation, which is when you change your nucleotide and you end up with the same amino acid, which is what we're going to see with changing GCU to GCA. And that's also going to be a transversion because we're changing this pyrimidine to a purine A. So we're going to do GCU into GCA, and we're still going to code for the amino acid alanine. So the protein's function has not changed at all. The next possible mutation we can go through is a read-through, which is when you change the stop codon into so that it codes for a different amino acid and makes the protein chain longer. So in this case, we're going to do UGA to UGG. So we're going to change this A, which is a purine, to a G, which is also a purine, which makes this a transition mutation. So this stop codon will no longer be a stop codon, and it will make the amino acid chain longer. The last mutation that we could possibly go through is a nonsense mutation, which changes a codon into a stop codon. So in this case, we're going to change UGG to UGA, which is another purine to purine change, which is another transition. So we're going to change that there, and that will stop translation into the amino acid and change the protein chain completely.